So welcome. This is the monthly lecture for the Atheist Community of Austin. And I've passed out some pamphlets to new people that I've noticed. If you're interested, you can hang around after the lecture and go join us at the Dog and Duck. We'll have lunch over there, and there'll also be an ACA board meeting. If you want to find out all the goings-on or not, then you can come over there. Okay, so ordinarily I would be introducing the speaker, and since I'm the speaker, I guess I'm going to have to introduce myself. I'm Steve Bratteng, and I've been teaching biology in one form or another since 1967. I recently retired from teaching at Westwood High School and now teach part-time at Austin Community College. And so what I'm going to present is some really basic information about how we play around with the parts of cells to make them do what we want them to do. So, many people that have had biology, maybe it was long enough ago that you don't remember all of the ins and outs of the chemicals that are involved. So I'm going to give you a little bit of background before we get into the biotechnology. So, as you see, about 95% plus of us is made up of just 4 elements: CHON, C-H-O-N, carbon, hydrogen, oxygen, and nitrogen, in various combinations, and there are literally thousands of different combinations that you can find in basically any cell in any person. Well, what happened? Okay, I don't know if you can see that, but this is a periodic table. And on this side, the 4 main things are kind of clustered together with similar electronegativities. Now, one of them actually is located over here, but its electronegativity puts it just this side of carbon. Now, what is electronegativity? It is the tendency for an atom to draw electrons toward itself when it's combined with another atom of some sort. So if it's combined with, for example, in the molecule of oxygen, 2 oxygen atoms, the electrons are equally distributed because it's the same element. But when you put oxygen with one of those other ones, the electrons will spend more of their time around the oxygen than they do around any of the other ones. And this will make a difference in the charge separation, so that one side of a molecule can be slightly negative and the other side can be slightly positive. This is the basis for many kinds of interactions. For example, water being a liquid at room temperature is due to the fact that it's a polar molecule. One side is negative, one side is positive, and so they hook up together in this hydrogen bonding scenario. Now, oxygen is significantly more electronegative than the other 3 main ones, and so whenever it's around, it's probably going to make that molecule a polar molecule. In other words, with a separation of the charges. And that's an important thing that we'll see. Okay, so in us and every other living thing, there are 4 major categories of chemicals that we can look at: carbohydrates, lipids, proteins, and nucleic acids. And I'm going to quickly show you what carbohydrates are: sugars and polymers of sugars. As you can see, there is carbon and lots of oxygen and some hydrogen. And so sugars are generally somewhat polar and are therefore easily dissolved in water. Now, you can join sugars together. Ordinarily, we would find glucose in this ring form, where this joins up with this carbon here, and it makes a ring compound. And you can have glucose subunits joined together in a long chain. One of these is starch, the one at the top, which we can use, and many other things can use as a food source. The one at the bottom is cellulose, which very few things can use as a food source, simply because the sugar units are flip-flopped here. They go every other. The enzyme that will break this down cannot do anything to that form. So this is the kind of thing we'll be looking at. The shapes of molecules are extremely important in how they interact with one another. Another important group, the lipids, is a very diverse group. It's mostly things that are somewhat nonpolar, are hydrophobic. Most of them do not dissolve well in water. Now, there is one important class, the phospholipids, that has a polar component. The phospho of the phospholipids is a phosphate group. It's phosphorus and 4 oxygens around it. So that makes one side of the molecule polar, and the rest of it is not. You use things that are similar to that when you wash your dishes, because detergents and soaps have a polar side and a non-polar side, and they dissolve in water, and they also dissolve in grease, and that helps you clean stuff. They also are the primary architecture of cell membranes. And now the cell membranes also have other things embedded in them, like important proteins that let things go through the membranes. Also, we have steroids. There are some important hormones that are in that class, as well as cholesterol. Now, cholesterol is an important substance in stabilizing cell membranes. Without it, cell membranes have a tendency to be very loose and will not stay together very well. And so it turns out that cholesterol is actually a critical part of your diet and of your cells. So a zero cholesterol diet may not be a good thing, because then your body has to make some of it. Okay. Oh, by the way, this unsaturated and saturated fats are the saturated ones are ones that have no double bonds along this chain of carbons. An unsaturated one has a double bond. Now, just recently, there's a move to outlaw trans fats. A trans fat is one where one of these hydrogens is on one side and one is on the other, a situation called trans. That's a situation called cis, C-I-S. What that does is it straightens out the molecule. And you'll notice that the saturated fat is a long straight chain, and when you put a trans fat in there, it acts like a saturated fat and packs together the same way saturated fats do. And it turns out that saturated fats are not that good for you.
True.
And therefore, trans fats are not that good for you. Okay, proteins. These are the guys that do everything in cells. Anything that moves is because a protein is making it move. They carry out chemical reactions by making them speed up to much faster than they would otherwise. And a protein is simply a chain of amino acids. There's an amino acid. It's got an amino group and a carboxyl group, which is an acidic group. And then it has in us and other living things, 20 different R groups. Those R groups you can see on the top there, those green boxes represent some of the R groups. And they have different characteristics, and therefore they interact with one another differently. And this is extremely important in how the protein will fold up and make a particular shape. And that shape will determine how it functions. Different enzymes have slightly different shapes and different affinities for molecules. And so if you were to change one of those amino acids, it might change the shape of the molecule, and therefore it might change the way it works. Now, where do we get the information to put those amino acids together to make things work? From nucleic acids. Nucleic acids, also a polymer, a long chain of subunits called nucleotides. Here we see 2 strands of DNA, one here and one here. Each nucleotide consists of a phosphate group, a sugar, and a nitrogenous base. And what we end up having is a backbone of sugar, phosphate, with that nitrogenous base sticking out to the side. And one nitrogenous base can bond with hydrogen bonds, kind of a weak bond, with another one in this manner. So, in a DNA or RNA polymer, where you have an adenine in a DNA, it will hydrogen bond to another chain that has a thing called a thymine. We usually abbreviate them ATCG. Now, if you had RNA doesn't have any T. It has instead of thymine, it replaces that with uracil. But nevertheless, it works the same way, where they have a very strong affinity to pair up in that particular way. Now, the sequence of those bases is a code. And this is our universal code. Virtually everything that we've ever studied has the same basic code of looking at 3 nucleotides in a row, which is what's called a codon. So like, for example, AAA would code for the amino acid lysine, and so on. And you see there's 64 different codes. Some of them code for, some different ones will code for the same amino acid, and some of them will code for only one. And so as the DNA is translated into RNA and then, transcribed into RNA and then translated into a protein, that sequence of bases in the DNA is the information to make all those thousands of proteins that are in us. Okay, so this is what happens. You have DNA, and there's an enzyme called RNA polymerase, which makes RNA. So it splits apart the double helix, copies one side, and makes a piece of RNA. Turns out there's non-coding stuff in that piece of RNA. So there's little bodies in there that snip it out and put it together. And so you end up with something called messenger RNA that ends up with a cap and a tail on it. Then it leaves the nucleus, and a ribosome latches onto it and starts reading that code. And as it reads it, it brings in another kind of RNA that has the specific amino acids attached to it. And so it reads along 3 at a time, brings in all of these transfer RNAs with their amino acids. They join up, it gets to a codon that says stop, everything falls apart, and you've got a polypeptide or protein chain, which then often has to be fixed up into another form to where it can actually do the work that it's supposed to do. We have 2 kinds of cells in the world. Those that have a nucleus, like ours, and then things like bacteria that do not have a nuclear envelope. And so that process of reading the DNA and making the protein happens all in the same spot. There's no regulation of the information exchange there. That's one of the reasons that we're a little more complicated than bacteria. Okay, then there's also some non-cellular genetic systems that we call viruses. They are a nucleic acid, either DNA or RNA, inside of a protein capsid, a coat surrounding them. And they will get into a cell. The nucleic acid will then elicit a response of making more proteins, more nucleic acid. And typically when these leave the cell is suffering and perhaps dead as a result. Okay, so that's our first 6 weeks refresher course of what's in cells and how it works. All right, so now we get into biotechnology. We're talking about evolution. Evolution is the reason that we have all of these interesting things in us and other living things. And they've been fooling around with their DNA and proteins and stuff for 3.8 or so billion years at least. And we recently realized, hey, they're doing some really cool stuff and we can take it from them and use it. And that's what we've been doing for the last 50, 60 years. We've been stealing stuff from, from bacteria, from other organisms, and from people and whatnot. We do that a lot. Okay, so we have a toolkit of some basic items that we can utilize. One of them are chemicals called antibiotics. Fungi and bacteria have been fighting each other chemically and otherwise for billions of years. And in the process, one of the chemical tools that they have are these things called antibiotics, and they can release these, and these will kill or impede the growth of other organisms. We use those, and now we figured out we could use these to fight bacteria back in the middle of the 20th century, and it was kind of a secular miracle, if you will, that we can now put a chemical in somebody and keep them from dying from a disease. Meanwhile, the bacteria that we were fighting have been evolving, and some of them are now resistant to all of those cool antibiotics, so we keep fighting them. Another weapon or tool is little circles of DNA in prokaryotic cells, and these are called plasmids. One of the main things that you find in most plasmids are genes that confer resistance to antibiotics. So this is now a countermeasure against the antibiotics that other organisms use. And so these little circles of DNA, and now if those are micrographs of little plasmids, by comparison, the large DNA molecule that's in every bacterial cell would probably be about as big a circle as this room is, like that, compared to those plasmids. Very small amount of information, but some of it very important. Question?
And those plasmids are transferred between different types of organisms?
They're transferred between completely unrelated types of bacteria. And that's one of the problems that we have, is that bacteria that are not related can have sex and get plasmids and thereby antibiotic resistance from completely unrelated organisms.
Scary
and
stuff.
even when a bacterium dies, its plasmids can be taken up by living bacteria. And have you heard of MRSA? Okay, well that's one of the things that we've managed to create by providing an atmosphere where they can evolve like crazy and do that. Another tool is thing called restriction enzymes. Restriction sounds a little funny, but what it means is an enzyme that chops up DNA. Bacteria have restriction enzymes, and there are dozens and dozens of different restriction enzymes that chop up DNA in a certain way. And they use this as a way that protects them from attacks by various kinds of viruses, because a virus will inject its DNA, and that takes it injects it. Well, they have a way of protecting their own DNA, and so when the virus sticks its DNA in there, it chops it up, and so it often protects them, although it's not a perfect defense like most things. Also, you notice that a restriction enzyme will recognize a particular sequence of bases on the DNA and cut in a certain way. We will be looking for restriction enzymes that cut in this kind of zigzag manner. If it cuts it straight across, it's really not very useful for our purposes. So we have literally dozens and dozens of different restriction enzymes, all cataloged away, so that if you want to cut a particular piece of DNA in a certain way, you look in your catalog and you find out the one that has a sequence that you're interested in, and you can chop that one. Okay, another one is TA, attack polymerase. TAC means Thermus aquaticus. It's a bacterium that was discovered in hot springs in Yellowstone National Park. It lives at temperatures that would kill most other organisms, which means that its enzymes are not denatured by that heat. If I'm sure you've boiled an egg. The egg white is mostly protein. You put it in boiling water or very hot water for a while, and it comes out as this opaque, solid mass. Guess what? It won't go back to being a liquid. So it's denatured. These organisms can live at temperatures that would cook many other things, and their proteins are not denatured as a result.
Are they found in those thermal vents?
Yes.
Yeah.
Yes, also. There is one, it's actually in a group called the Archaea. It's not a bacterium. It looks like one. Its optimal temperature is 106 degrees Celsius. 100 degrees is the boiling point of water, and it can survive up to 120 plus degrees Celsius.
But those vents are like 700, aren't they? Because of the pressure?
I don't know if they're 700. I don't know a value for that, but it's too hot for me.
Yes.
In any event, we can use enzymes from organisms like this and heat them up and cool them down. We'll see how that works in a little bit. And finally, in our toolkit, we have an enzyme called reverse transcriptase. The process of copying DNA to make RNA is called transcription. The process of copying RNA to make DNA is reverse transcription. Things like HIV and other retroviruses carry along a copy of the enzyme reverse transcriptase. And when an HIV particle inserts its RNA into a cell, that enzyme makes a piece of DNA, which then goes into your nucleus and becomes part of your DNA. And then it gets read and it makes more proteins and more RNA and stuff, and when it leaves the cell's dead, basically. And anyway, okay, so we find out we can use those for stuff. Now, our stem cells have a little repair kit that has some reverse transcriptase. Probably the retroviruses stole it from us in the first place. Okay, so what we'll be looking at is making recombinant DNA. Recombinant DNA is a piece of DNA that has sections from different organisms together in one thing. We'll find that different ways of making that are part of genetic engineering, and ultimately we want to have a situation of biotechnology where we can use that to benefit us in some way or other. Okay, cloning. Cloning is making identical copies of something. It could be a whole organism, or it could just be a piece of DNA. And we often use plasmids in making clones of particular genes. And then you can put the plasmid that has the gene into a bacterial cell, and it will grow like crazy, and you will get many, many copies of that piece of DNA. And so, well, that's what I just said. All right, so here's a slide outlining how this works. You have a bacterial cell that has a plasmid in it. You have another source of DNA. It could be a human cell. And you cut out a piece of DNA using restriction enzymes, and you cut the plasmid, and that DNA will get stuck in there. And then you've got these plasmids that you can grow up in these cells, and you can grow them in these gigantic vats, and you can make literally hundreds of pounds of whatever it is that you're interested in making. And they have been doing that, and you can see a few of the applications. They use it to put pest resistance in plants, to break down toxic waste, to make things to human growth hormone and other kinds of applications. Okay, so to a little more detail on how we do this, using restriction enzymes to break down DNA from different sources. What we want to do is get sticky ends. And this is what a sticky end is. This and this are complementary pieces that when they are broken apart by the restriction enzyme, they will match up with those same ends regardless of what source of DNA. So you can chop up plasmid DNA with this restriction enzyme, cut up another organism's DNA with the same restriction enzyme, and they will have matching sticky ends. So you can just put them together and put in an enzyme that ties them together, a DNA ligase, and you will end up having recombinant DNA. This is how that works. So we have a restriction site that we've identified in two different sources of DNA. We break it apart, we get sticky ends. We have another source of DNA in gray that has been chopped up using the same restriction enzyme. It had a restriction enzyme that cut at one end and at the other end with the same enzyme. Put them together and you will get that. Now sometimes they just go back together the way they were, which is not terribly useful. So you have to have a way to screen to get the ones that are actually recombinant. And we'll do that. So there we have a recombinant DNA molecule. Question?
Are you going to go over how they screen to get the useful bits out?
Well,
okay.
Briefly. Okay, so here's a more detailed example of what we do. Or I don't do it. Okay, this is a bacterial plasmid made out of DNA. It has two important genes on it. One of them has a restriction site in it, a place where a particular restriction enzyme will cut it. The other gene is a gene for resistance to ampicillin. And this is part of how we will end up screening to find where the thing is that we want. Now, the gene that has the restriction site in it turns out to be a gene that allows the bacterial cell to break down lactose. So, in this case, for some reason or other, they picked hummingbird DNA to chop up using the same restriction enzyme that they cut the plasmid with. So you end up with a bunch of pieces of hummingbird DNA and you mix them in there. In some cases, you will get the gene that you're interested in stuck in a plasmid. And then you can put those into cells and you can then grow them on a petri dish, on a nutrient agar. And you can see the ones that do not grow where there's ampicillin. They're going to have a plasmid in it. And if they cannot utilize lactose, then you know that they've been cut in the middle of that lac gene. You can also then make a probe that will then specifically identify that particular gene, because you already know some of the sequence of it, and we'll see how they make a probe in just a bit. Also, well, that plasmid is called what do we call it? We call it a cloning vector. Natural plasmids usually can only carry an extra 1 or 2 genes, so we make artificial plasmids. Bacterial artificial chromosome is a plasmid that we manufacture that has many more genes in it. We can put, a dozen or more genes in one plasmid instead of just 1 or 2 in the ones that the bacteria make.
See, when you open that circle to put that new DNA in there, do you have to get rid of those original ones that are in there, or do they just break?
No, there's no original ones in there. It's a break.
Yeah.
It's something that is, so that original thing is now in 2 pieces.
Okay.
And that's why it doesn't make the enzyme for breaking down lactose, because what you've done is you have disrupted that gene by putting another piece of DNA in there, and it won't work anymore.
Okay, I see.
Okay?
So basically the way you screen it out is you just culture a whole bunch of everything that results.
Yeah, and the ones that have certain properties are the ones that you then know have the plasmid that you're interested in them.
You just keep those and discard the rest. Basically.
Or save them. Yeah, you never know what you might use them for. Anyway, so here is a really important technique. It is called the manufacture of complementary DNA. And this is exceedingly important. Now, remember that process whereby DNA is read, made into RNA. RNA is processed, and then it's messenger RNA, and then it goes out in the cytoplasm where it gets read by ribosomes to make a protein, right?
Got it, yeah.
Okay, that original DNA had non-coding parts in it, which are now no longer in the messenger RNA. We have that enzyme reverse transcriptase that will take RNA and make it into DNA. So what we do is we reconstruct the original gene without the non-coding parts, without the introns in it. Now, if you had put the original gene into a bacterial cell, it wouldn't work, because the bacteria do not have a way to remove the non-coding parts, what are called introns. But if you take those out, you can now put a piece of DNA into a bacterial cell, and the bacteria will treat it as if it were one of its own genes and will read that information and make whatever protein that is. Okay? And it turns out then, all the complementary DNA that you make is DNA that was actually read and expressed in that cell. In any given cell, only a minute portion of the genetic information is read at any given time. And so if a particular gene is turned on, then you will find its corresponding messenger RNA in the cytoplasm. and not others that are not turned on. And this turns out to be very important in figuring out some stuff. So this is how we do it. We extract the messenger RNA from the cytoplasm. This is done, you grind up the cells and you centrifuge them, and the messenger RNAs will be in a certain band and you can take them out. Okay. All right. So you use that enzyme, reverse transcriptase, that you stole from the viruses, the retroviruses, and it will make, eventually, cDNA, complementary DNA, which you can then use for various purposes, like putting into a plasmid or whatever you want to do with it. But that complementary DNA is a very handy substance to use. Okay, now if you want to find a specific gene, if you know at least some portion of its code, you can make then a complementary piece of DNA, or actually RNA, something complementary that will match up. So, for example, we have C's always match with G's, T's with A's, C, G. So you make a complementary piece of nucleic acid, and you can use radioactive materials to create that probe, or you can tag it with a fluorescent dye. Either way will work for finding it later. The radioactive materials was the way they did it to begin with, and now they're moving more into using fluorescent dyes as much as possible. It's a lot less dangerous working with fluorescent dyes than with radioactive materials. So, now by the way, this nucleic acid hybridization, this simply means that if you have two pieces of complementary nucleic acid, they will come together when it's cool enough. If you heat them up, they will go apart. Hybridization, they will join together. So you can put some little pieces of DNA on in a thing with other DNA, and they will find the matching part and stick to them. And then you actually have this automated process that will chop up DNA, run it through an electrophoresis apparatus, and then deposit it in little wells. These are little plates, and they have holes, and each little hole from this automated process will have a different piece of DNA, different genes in it. Now, you want to find the one that has the gene that you're interested in. Which well is it in? Okay, so you put this membrane on top of it, and then you have the probe there. It will actually pull off one of the strands of the double helix. And then you treat it with the probe, and it will find that gene and latch onto it. And if it's radioactive, then you put photographic film on it, and it will expose it right where that probe was. So then you know exactly which well had the thing you're interested in. So the next time you run that, then you know, okay, I want to get that, and you can find that gene every time you need it. Okay, so the term expressed, when it comes to genes, means that the DNA is being made into RNA, which is then being read and made into a protein. That gene is turned on. It's being expressed. Okay, now, in bacteria, well, in all bacteria and eukaryotes, the thing that makes the RNA polymerase, looks for a thing called a promoter. It's a little section of the DNA that says start here. And they're different in prokaryotes and in eukaryotes. It's a much more complicated system in eukaryotic cells. So if you put human DNA into a plasmid and then put that into a bacterium, if you use the complementary DNA, then you don't have a problem, because it will, most of the time, it will find the promoter because you're using bacterial DNA plus, eukaryotic DNA, and it will find the prokaryotic promoter. So there's a lot of these little subtleties of how to make this work the way you want it to, and this has taken years of fooling around doing things, and eventually you have a protocol established For each particular gene that you're interested in, there's a certain protocol for how to deal with that. And if you're interested, you find a new gene that you're interested in, then you have a few weeks to a few months of puttering around to find the system that works best to get that one and express it. I've got a lot of stuff. Oh, there is one thing here that when you've got these plasmids, how do you get them into another cell? Well, if you're trying to get them into a eukaryotic cell, it's a little difficult because they don't like to do that. So what you do is you zap them with a little electric charge and they go, ow! And the plasmid slips in there while they're not looking, so to speak. Now, you can also inject plasmids into an animal cell, into an egg, for example. Now, with plants, they developed a thing, it was really cool. The first one of these is called a gene gun. And what they did was they had the plasmids in a solution, suspension, and they coated little microscopic gold BBs with the plasmids and put it on a membrane on a .22 caliber cartridge and actually shot at the plant with the .22 gun. They now have they have a more sophisticated way of doing that with using a vacuum thing. But anyway, they actually had a .22 caliber cartridge, and they would shoot at it, and then the little BBs would go into the cells through the cell wall, deliver the plasmids. Of course, some of the cells were destroyed by the plasmid, but some of them, a couple of BBs would go in there and deliver the plasmids, and you only need one to work, really. Once you got that, then you can grow up a lot of cells, and in fact, you can grow an entire plant from one of those cells. Oh, how are we fixed on time? Because there's a little movie here.
It's 5 till 1 right now.
Okay. There's a movie here about putting genes from one kind of organism into another. Maybe it'll work. Okay. Well, it's not doing it. It's set. Oh, there we go. It just has to think about it. No, it's thinking about it. Crap.
Navigation to the wallpaper.
Turn out. I have a few files here. I saw one that said Biotechnology, and if I could just stop on it. Well, maybe I'll pantomime it.
Just do a soft shoe and
You may want the internet.
It's looking. It's got. I had the file actually downloaded, and
okay.
It's not doing it. I'm not sure why exactly, but it's not doing it.
some kind of gypsy curse.
Thanks for all the useful information. Okay. So this video, it has to do with something called Hox genes or homeobox genes. These are genes that they don't make a protein product like, an enzyme or anything. What they do is they regulate many other genes and turn them on. And you have different Hox genes have different functions. And there's a Hox gene that was found that causes fruit flies to make their eyes. And a similar gene was found in mice. And so what they did was they said, well, let's take the gene out of the mouse and put it into a mutant fly that is called eyeless. That gene is defective. So they put the mouse gene, injected it into fly eggs, and the flies had normal fruit fly eyes made. So that gene just says make an eye. It doesn't say make a mouse eye or anything. It says just make an eye right here. And if they have the genes to make a mouse eye, they make a mouse eye. And if it has fruit fly eyes, it makes fruit fly eyes. Now, one thing that they didn't tell you in this little video, if we had seen it, was that they actually don't have normal fruit flies produced that way. They have eyes all over them. They got eyes everywhere because they just injected it into the egg. It wasn't in the right place, and so you end up with these goofy-looking flies that have eyes everywhere. And they don't care. It's just fruit flies, right?
Well, fruit flies today, tomorrow it's dogs or something.
Yeah, or something. Well, so that's what we're talking about there. These Hox genes that turn on many other genes, and they're actually organized in a linear sequence, and they do the different parts of the body in that sequence. And you can move them around. They've put the gene that is responsible for making a leg into the head of a fly, and they will then have a leg coming out of their head instead of an antenna. Tricks to play on
So these are mostly used during development, is that right?
Yeah.
So you're really talking about embryology, kind of.
Yeah, that's where, there's such a lot of stuff going on. You have one cell that has all the information, and then over time you get millions and millions of cells. Each one is only using some portion of all that information. And how they sort out who uses what is what embryology is all about. And part of it has to do with things that will turn genes on and off, allow them to be expressed or not be expressed. All right. The polymerase chain reaction is a technique created by a Californian surfer dude named Kerry Mullis, who won a Nobel Prize. Now, he was such an odd duck that it was not clear that even though this PCR technique is now used throughout the world, virtually every molecular biology lab will use this if they need to make more DNA from a source. He apparently dropped a lot of acid and smoked a lot of pot back in the 60s and did a lot of surfing. But one night he was driving along and he had this great idea and remembered about those enzymes from the hot springs that function regardless of how hot it gets, basically. So he came up with this technique whereby you have a small piece of DNA that you've gotten from some source. Like you could find a hair, you could find just any little bit of blood that has DNA in it. You can take that DNA and then make thousands and thousands of copies of it using this technique. It's like a photocopy machine for DNA. And it's the polymerase chain reaction. And it works by putting all the ingredients in there, including the DNA polymerase that doesn't get destroyed by heat. You heat it up, you cool it down. You heat it up, you cool it down. And every time you do that cycle, it doubles the amount of DNA. So that in a matter of a few hours, you can have enough DNA to do all kinds of tests on it to, for example, identify somebody based on their DNA. Okay, the kinds of things that we can do with the DNA, that PCR technique is another way of cloning. Now, it's a lot faster and in some ways easier than using plasmids and sticking it in bacterial cells. It's just, for very large amounts of DNA, it's not that reliable. So that's why they still use the plasmid cloning technique a lot. So we can compare genes and alleles. Alleles are different forms